Here is a SARS coronavirus 2, commonly known as SARS-CoV-2. It is the cause of COVID-19. It is represented floating in the thin layer of mucus that covers the surface of our airways, from the nose and mouth to deep inside our lungs. Sticking out from the spherical surface of the virus are spike proteins, about 20 to 30 of them on each virus. These spike proteins are complex pieces of virus machinery designed to allow the SARS-CoV-2 virus to release its contents into an airway cell below the layer of mucus. Inside the virus are two strands of RNA, each of which provides the instructions for an infected cell to make and assemble many thousands of daughter viruses. Each virus can be thought of as a message in an envelope. The strands of RNA are the message, and the outer layer of the virus with the protruding spike proteins is the envelope. The task each virus is designed to achieve is to attach to the surface of a target cell, merge the virus envelope with the outer surface of the target cell, and release the virus RNA into the target cell, which will then make thousands of daughter viruses, and then on and on, repeating the process. The spikes projecting from the envelope of each SARS-CoV-2 virus are composed of three identical proteins, which will clump tightly together to form the spike proteins. Each of these three identical proteins is folded into a complex structure with four long, straight portions linked by hairpin bends. Three spike proteins clump together to create the 20 to 30 spikes on the surface of each SARS-CoV-2 virus. The receptor binding domains at the tip of each spike protein are able to swing on their stalks, either to extend away from the virus envelope or to rest on the top of the spike protein. The target for the SARS-CoV-2 virus is the ACE2 enzyme, embedded in the surface membrane of respiratory airway lining cells. Each ACE2 enzyme molecule on the surface of airway lining cells is always accompanied by a TMPRSS2 enzyme. A SARS-CoV-2 virus floating in the mucus lining, the surface of the respiratory airway, has come close to the surface of a respiratory airway cell. A single receptor binding domain has bound tightly to the ACE2 enzyme. The tight binding of the receptor binding domain to the ACE2 enzyme leads to minor changes in the shape of the spike protein. This allows the TMPRSS2 enzyme to snip the spike protein. The virus is no longer directly bound to the surface of the airway lining cell. Another enzyme, furin, floating in the mucus, now attaches to the portion of each spike protein that extends beyond the last straight portion. Furin snips off the tips of each of the three spike proteins. Once this has been done, the next stage of invasion of the target cell by the SARS-CoV-2 virus can begin. The three shortened spike proteins now unfold. At the tip of each shortened spike protein is a portion that can bury itself firmly in the surface membrane of airway lining cells. Once the tip of each shortened spike protein is inserted into the surface membrane of an airway lining cell, the SARS-CoV-2 virus is firmly attached to the airway lining cell. The spike proteins will now pull the SARS-CoV-2 virus closer to the surface of the airway lining cell. The three spike proteins refold, but with only one hairpin bend, This refolding pulls the virus down towards the surface of the airway lining cell. The refolding of the spike proteins brings the envelope of the virus very close to the surface membrane of the respiratory airway cell. When the envelope of the virus touches the surface membrane of the airway lining cell, these two membranes fuse, creating a channel through which the virus contents can flow into the airway lining cell. The process from attachment of the virus to the ACE2 enzyme to entry of the virus RNA into the cell takes about 10 minutes. The entry of the virus RNA into the cell infects the cell, and within about 10 hours, many hundreds of viruses will be released from the infected cell. The cells of the immune system respond quickly to the virus infection. Within about 5 to 10 days, immune cells will have produced large amounts of antibodies that can bind strongly to many different parts of the spike proteins. These antibodies are released into the blood, the mucus in the airways, the mucus in the intestines, and into tears and breast milk. Antibodies that bind strongly to the receptor binding domains at the tip of each spike protein prevent them from binding to ACE2. These antibodies neutralize the virus. There are several different types of COVID-19 vaccines, many of them similar to the safe, effective vaccines that have been used worldwide to prevent other viral infections for very many years. Inactivated vaccines, such as the Chinese Sinovac vaccine, are one type of vaccine that may well be widely used. A very simple way to make a COVID-19 vaccine is to grow SARS-CoV-2 viruses and then to inactivate the viruses by damaging the RNA message in each virus. These inactivated viruses cannot reproduce in a vaccinated person, but do stimulate immune responses against the virus envelope proteins, including the spike proteins. Inactivated virus vaccines in widespread use include the polio and hepatitis A vaccines. Subunit vaccines, such as the Sanofi Pasteur and GSK vaccines, are also in development. A more sophisticated way to make a COVID-19 vaccine is to genetically engineer a yeast or some other cell so that when that cell is grown, it releases large amounts of SARS-CoV-2 spike proteins, which are then collected and purified. Alternatively, the cell may be engineered so that it releases only the receptor binding domain of the spike protein. These vaccines have no genetic instructions and so cannot reproduce. Subunit vaccines in widespread use include the hepatitis B and influenza vaccines. Attenuated vaccines, such as the Indian codogenic vaccine, are also under development. It is possible to modify the RNA instructions within a SARS-CoV-2 virus to make the virus weaker. Attenuated viruses can multiply in a vaccinated person, but have almost no chance of causing disease. Attenuated viruses stimulate immune responses against the virus envelope proteins, including the production of neutralizing antibodies to the spike proteins. Inactivated virus vaccines in widespread use include the polio and hepatitis A vaccines. Viral vector vaccines, such as the Oxford vaccine, have also been developed. It is possible to take a completely different virus that can briefly infect people without causing disease and replace the RNA code for its own spike protein with the RNA code for the SARS-CoV-2 spike protein. When the hybrid virus multiplies, it will insert the SARS-CoV-2 spike protein into its envelope. This hybrid virus can be used as a replicating viral vector vaccine or an inactivated virus vaccine. hybrid viral vector vaccines are very similar to the extremely safe and effective vaccines used to protect people against Ebola. The first vaccines to become available against COVID-19 are RNA-based vaccines, such as the Pfizer and Moderna vaccines. It is possible to manufacture the portion of the SARS-CoV-2 RNA that provides the instructions for making the whole spike protein, or that provides the instructions for making the receptor binding domain. But this RNA provides instructions for no other parts of the SARS-CoV-2 virus. This RNA enters the cells of the vaccinated person, and these cells then produce large amounts of whole spike proteins or receptor binding domains. The immune system responds to these proteins. This is a new technology, not previously used in human vaccines.